Our last presentation of the day is from the Allen Discovery Center for Cell Lineage Tracing, and the work will be presented by Shendure and Long Kai.
Okay, thanks, Kathy. And so I'm presenting on behalf of the center. As Kathy mentioned, I'm going to give half of the talk and then Long the other half. And this center is split between UW, Harvard, and Caltech, and co-led by myself and Michael Elowitz. So I'll just start maybe by a quick acknowledgement of the impact that the foundation has had on our own work. This is a topic that my lab did not work on at all 5 years ago when I got basically a solicitation to apply for something in this area from the foundation. And now it's the vast majority of what we do. And moreover, these meetings is really where this group really came together a few years ago and put the idea for this center together. So okay, so the problem that we're trying to tackle, just as a brief introduction. Let's go back here. Okay. So the problem we're trying to tackle is how to globally record cell lineage and ideally everything that happens during the course of an organism's development. So the only organism where we're close to this is, of course, C. elegans, famously mapped by John Sulston. But for basically every other organism where we might want to do this, the challenge is much more difficult due to the lack of transparency, variable developmental outcomes, and also simply the scale of the problem. So what we're trying to do is really to take advantage of the proliferation of amazing technologies over the last few years that allow us, I think, to take a very different approach to tackling this challenge than what John Sulston did through visual observation. And so, if you try to just boil down the questions that we're trying to ask through these approaches, they can boil down to how are cells related, lineage, right? So knowing how each cell is related to each other cell, knowing what each cell is in that lineage, what its role is, or what its type is, or however you want to call it. And then knowing where each cell is located, ideally both in space and time. Okay, so I'm going to focus, so first, as a preface, I think our entry point to this was the development of independently by our group and Michael's group and Long, as well as a few other groups of technologies that rely on CRISPR genome editing as a means of recording information. And so these were published a couple of years ago now, so I'm not going to talk about these in any level of detail, but they're complementary technologies. And now I think what we're trying to do is to build out the rest of kind of the other technologies that are necessary to marry with these in order to really go for the global, spatially and temporally resolved lineage of vertebrate development that we would like to achieve. Okay, so I'm gonna focus on kind of a technology that allows us to access kind of a mix of these first two questions. How are cells related, and what do cells do? What are the various cell types? Okay, so single cell methods that I think most people in this room are probably aware are rapidly evolving at an exponential rate over a relatively short span of time. So the particular flavor of technologies that our group has developed over the past few years are based on this concept of split pool indexing. And the idea here is that, and I'm not going to go through it in all of its detail, but the idea is that you are effectively uniquely labeling the nucleic acid contents of a large number of single cells without ever physically isolating each cell. And the way that you do this is to split the cells out to some number of wells, perform some kind of in situ reaction, such as a reverse transcription, let's say, with a bar-coded primer, pool the cells, split them back out again, do another round of labeling. And the idea here is that as you accumulate additional rounds of labeling like this, the number of cells that you can uniquely mark grows exponentially. And so over the past couple of years, we've developed various flavors of these SCI technologies for looking at single cell ataxy, so chromatin accessibility, like Jason talked about, nuclear architecture, DNA sequencing, RNA sequencing, methylation, and most recently co-assays. We're able to measure both chromatin accessibility and expression in each of many single cells. And it's really all just this single paradigm of split pooling of cells or nuclei and coupling that to various tricks of molecular biology. Right? So So what we've been trying to do over the last year or two with this technology is to build various kinds of atlases of molecular state for key model organisms. So here, for example, the worm, where we're able to measure 40,000 transcriptomes for an organism with at this stage, only 750 cells or so. And so we've kind of over-covered effectively the cellular content of the organism. And so some of these clusters actually correspond to single neurons within any single worm, right? And they appear as a cluster of cells because we've over-covered the content. We've also tried to do this with some degree of temporal resolution. So here looking at fly embryogenesis and chromatin accessibility, and seeing how that evolves as cells go through the initial formation of germ layers. And then more recently trying to, at scale, bring this into mammals. So looking at chromatin accessibility in 100,000 cells in the adult mouse, and trying to intersect that kind of information with studies of human genetics of common disease. Okay, but what I'm gonna focus on today is some unpublished work involving trying to take this from 2-level indexing, so 2 rounds of split pool, which is what we did previously, to 3 rounds, which is where the power of this, I think, really kicks in. And so Jun Cao, who's the student who developed this, put together this protocol for 3-level indexing, and with it was able to profile in about a week and a half of physical work from one individual, was able to generate libraries corresponding to 2 million cells of single cell data. And then we were able to sequence this on 1 run of Illumina NovaSeq, right? And so if you do the math, it boils down to about $50,000 of direct cost for 2 million cells worth of data, which is substantially cheaper than alternative technologies. And so the problem that Jun applied this to, together with Malte Spielman, who is a visiting scientist, is looking at mouse development, specifically this period of 4 days between E9.5 and E13.5, which is where a lot of exciting things happen in organogenesis. One of the nice things about these Sky protocols is that you can actually encode different samples to each well during the first round of indexing. So we're basically putting individual embryos or cells from individual embryos into each well, and then performing this as a single experiment. And this has the effect of at least mitigating some of the batch effects that you might expect if you were running these serially. And so here we're doing 10 to 15 replicates from each of these time points at 9.5, 10.5, and so on and so forth. So yeah, this is just I'm going to go through this quickly. This is showing scale. So here we're really going for breadth, a very large numbers of cells, but sequencing them at a pretty shallow level. So only about 500 UMIs, or unique molecular identifiers, per cell. We could double this probably through deeper sequencing, but probably would require more optimization if one wanted to get a substantially larger number of transcripts per cell. Okay, so yeah, so the 2 million cells are split across these replicates from these time points, and then we can do the usual things. So this is a t-SNE plot where each point, there's 2 million points here. Each point reflects 1 cell. And you can cluster these into groups. You can then annotate these clusters. I think one important point is that if we superimpose cells derived from from the same time point, replicates from the same time point, they're virtually indistinguishable in these kinds of plots, suggesting that we did successfully mitigate any batch effects, or at least the ones we would mostly worry about. So we can do the usual thing, and based on the literature, annotate these clusters. And so we're able to identify 38 major groups, and then we can take each of these clusters, recluster them, and try to annotate those. And in total, we have about 550 of these clusters that are transcriptionally distinct subsets of cells that we're now, in the very painstaking throes of trying to go through and annotate. So just to dive a little more detail into one example, this is the apical ectodermal ridge, which is a well-studied system in limb development, where this thin layer of epithelia sits on top of the developing limb bud. And it's a transient population. It appears and disappears. So we expect it in this window, but we don't expect it to be that many cells. And so can we find it? So it's well-studied, so we know where to look. So we can pull out one of these clusters, which corresponds to epithelial cells. We can recluster those cells, and sure enough, this little nubbin down there has a lot of cells that express a specific marker, FGF8, for the apical ectodermal ridge. And so there's only about 1,200 cells, which is a small fraction of our data set out of 2 million, but that's about what you would, on the order of magnitude of what you would expect. So is this really the AER? So one thing we can do, these are sparse data at the single cell level, but we can reaggregate the cells within any given, within this cluster. And then we can look at other genes that are specific to the apparent AER and perform whole mountain C2s. And in fact, every marker that we've tested so far does in fact be specifically hybridizing to the AER there, which is shown right there. All right. Okay. Well, let's see here. So one of the limitations of t-SNE as an algorithm for people who've worked with it is that it doesn't, it does a nice job of visualizing data sets like this, but doesn't necessarily place related cells one near one another. These are various subsets of neurons that are kind of all over the place, right? And oligodendrocytes are just CNS cells in general. So Cole Trapnell, who's a part of our Allen Center and we work very closely with in this area, his lab developed a newer version of their tool Monocle, which relies on a very different set of operations than I think what has been done in the past, and in particular takes advantage of an algorithm called UMAP, which was developed by McInnes and Healy rather recently, and has very different properties than t-SNE, and in particular is better about putting related cells near one another when you map them into these spaces. And then Monocle proceeds to basically reconstruct trajectories through this space. So here's a slide. So this is a fun animation that Cole made where we're taking this 2 million cell t-SNE and transforming it into a UMAP. And you can see here how different they're going to look. And so before we had 38 clusters that were all kind of stuck together. Now we're in 3 dimensions, which is pretty necessary to actually separate this out. It's also a lot more fun to view. And you can see the data at some level appears less complex in the sense that it's grouped out into these 10 or so major trajectories, we'll call them. And these major trajectories correspond to the kinds of things that you might expect, but with the related cell types near one another, right? So here just walking through, I think most of them, we have endothelial cells, hepatocytes, mesenchymal cells, epithelial cells, hematopoietic cells, and then basically all of the CNS. And then some of those other little guys there are various neural crest subpopulations. And so, one of the reasons this is not all connected as a single rooted trajectory is that we started at 9.5, right? So the data set is missing cell types that might correspond to things that relate some of these major trajectories to one another. Okay, so in a nutshell, we went from 38 of these, quote, major cell types with t-SNE to these 10 kind of supergroups that are, that correspond to what we would largely expect with developmental biology in terms of their coherence and how the t-SNE clusters map to the Monocle 3 clusters or trajectories. So just as kind of a more detailed zoom in here, the other piece that's it's not completely reflected in the coloring here, which is of the clusters from t-SNE, but we can really see the time axis here. So down this elbow of this supercluster or the super trajectory are radial glia cells, and then these project out in one direction towards increasingly mature neurons. But if you were to go now in this other direction, they project out towards oligodendrocytes, right? And if we were to map time onto this, right, E9.5, 10.5, there would be a focus of E9.5 cells down here kind of going in both directions towards later time points. So this is, there are other ways of viewing this data, and we can walk through and try and annotate various aspects of at greater detail trajectories within these super trajectories, so to speak, right? So that was neurons here. We're looking at various endothelial trajectories, mesenchymal trajectories, and then neural crest subtrajectories. One of my favorite ones, I'm not sure where it's in here, but there's a subpopulation of 88 cells, which out of 2 million, which correspond to the primordial germ cells, right? So they're just extremely rare, but also very transcriptionally distinct from everything else. And then this is an example of taking one of these plots where now we're just looking at epithelial subtrajectories, but we're coloring it by time. And you can see these densities of early cells from E9.5 that project out in different directions in kind of a fashion that's consistent with the temporal order towards populations of E13.5 day cells of various types. So just different developmental differentiation trajectories, including some here like the apical ectodermal ridge, which projects out of the surface ectoderm and then back into the epidermis, right? Okay, and here's just one, this is just one more example of the kinds of ways we can view this data and trying to connect it back to actually getting more insight into developmental pathways. This is just pulling out all the cells corresponding to muscle development. We can then turn off the lights and then color this by gene expression of various markers of muscle development, including early markers and later markers. And the main point I want to make here just is that we can use these kinds of data to look for not just trajectories, but also patterns of branching and convergence that might shed light onto different pathways that cells can take, including for ending up at the same endpoint, right? By virtue of starting at different places and also taking different paths, right? Okay, so I'll stop there. That muscle data I showed is just that little nubbin right there, and so I think we've scarcely scratched the surface of what one could do in terms of trying to mine all the data that's here, but this should be available hopefully within a month at our website. And I'll stop there, and I think I'm going to hand it to Long, who's going to cover this second part.
Thanks, Jay. So Jay, how do I get out of this?
Okay. I forgot to transition you.
Okay, here. Thanks. All right, so I'm Long Kai. I'm representing the Caltech part of the Allen Discovery Center. So we heard yesterday from Allen and a lot of speakers today that the imaging approach has the potential to scale to the genomic level. And I want to convince you with this talk that actually we're already there. Okay, so what we're looking at here is in a single nuclei, we're looking at about over 10,000 transcription active sites are painted. Each one of these dots correspond to individual active sites. We know exactly what the active site is. And so this is actually looking at the nascent transcriptome of the cell. Everything I'm going to show you is done on commercial confocal microscope. So no fancy optics. So how do we do this? Well, so we developed this technique called seqfish, or sequential fish, where we use multiple rounds of hybridizations to barcode a large number of transcripts. And the way it works is that we can hybridize against the same set of transcripts over multiple rounds of hybridizations, such that each time the dots, even though they come back in the same positions, they switch colors. So for example, here, mRNA1 starts off in green channel, and then on the next round of hybridization, it ends up in blue, and mRNA2 starts in red and goes to green, and so on. So this way, by doing multiple rounds of hybridizations, even with a few different colors available in regular microscopy, you can scale up to a very large number. So you can see with 5 colors available, let's say, just 3 rounds of hybridizations allows you to get to 125 different barcodes. With just 6 rounds of hybridizations, you can get to essentially the transcriptome level. And I remember the first time I talked about this at the first Allen meeting in San Diego. I think I showed the preliminary results back in 2014 of only 12 genes. I'm going to show you that actually now this number is entirely doable. But before that, what we've done is we applied this technology to looking at hundreds of genes in mouse brain slices. So here you're looking at one piece of a part of a cortex. We're looking at layer 1, layer 2. And then if you focus on this region here, over multiple rounds of hybridizations, these dots are the same over 4 rounds of hybridizations, but they switch colors. So for example, this purple dot goes from purple to red to yellow. And you go back to that barcode lookup table, you know exactly what that gene is. And so you can then segment individual cells from the images and then count how many times a particular barcode occurs. That gives you the copy number of that particular transcript. And you can take the single cell data, you can cluster them, and this is how we get images like this, where we have individual cells showing these blobs, And then this is actually in the mouse hippocampus. We can see there are astrocytes based on the expression levels of hundreds of genes, and then there are excitatory neurons inside this pyramidal layer. And I would say that this work is actually probably still the best protocol in working with mouse brain slices. And in fact, if you look at the number of barcodes we can call out per cell, it's on the order of 3,000 or 4,000 per cell, which is actually about 10 times more than even some of the newer methods that came out recently. And this is all because single molecule FISH, which is the technology it's based on, is highly efficient in detecting RNAs. We think we're detecting basically all the transcripts with something like 80–90% efficiency in this multiplexing. So a couple years ago, what we decided to do is look at not just the messenger RNAs, but also the introns. And the motivations for doing this was that introns are very short-lived. They usually are co-transcriptionally spliced out and degraded in the cell. So when you look at introns, you're essentially looking at what the cell is trying to do at that moment. Whereas if you were looking at mature RNAs, we're looking at accumulated information in the last couple of hours. So this way, we can look for information that are dynamic and not smeared out by the more long-lived mRNA measurements. And then, of course, in context with memoir and recording, by scaling this up, we can also, scaling this method up, we can also show that we can read out many, many barcodes at the same time in the cells. Okay? So this experiment was targeting now over 10,000 different transcription active sites. Basically picked the first introns of these genes. These genes were essentially chosen randomly. We could have done all 20,000 genes. It was just a matter of the cost of purchasing the probes. So just to give you a sense, there was about 25 probes per gene. So overall, there was 250,000 probes purchased. It didn't cost that much, actually. It was only about $15,000 or so, and it's a one-time cost. So you hybridize the probes onto the cells, and here you have individual mouse ES cells and the nuclei. So you can see that each one of these dots correspond to a single transcription active site, where there's nascent transcripts coming off of transcription assembly and is still bound to the chromosome. And the holes in the cells essentially correspond to nucleoli, or transcriptionally inactive regions. And if you zoom in again to this little box here, we can see that over multiple rounds of hybridizations, that changes colors, so you can decode exactly what that transcription active site is and what the genomic loci is in that particular cell. So you can do this essentially for all 10,000 loci. So I'll show you the reconstruction in a second. You saw it in the beginning already, but also, and after we've done those 10,000 intron multiplexing experiments, we can perform messenger RNA measurements. And typically, we'd look at, on the order of 20 to 60 mRNA molecules, depending on the experiments. So here, showing the two pluripotency factors. We can look at link RNAs in the same cells. Here, mallet and fire, marking out different nuclear bodies. And at the very end, we can also do antibody stainings, right? So basically, we can do these kind of multi-omics measurements going from introns to messenger RNAs to proteins. And I think the difference is really just a time scale of the information stored in each of the molecules. Introns are very short-lived, mRNA is sort of in the middle, and proteins are very long-lived. I think that's what's giving rise to the sort of lack of correlations between some of these measurements. It's not really that the proteins or the mRNAs are very different from each other. It's just that you're talking about different time scales. Okay, so as promised, this is the reconstruction. If I can get this to play. So you're looking at a single cell, and the white parts are the nucleoli, and the blue are the heterochromatic regions. And then, of course, each of the dots is a transcription active site. And then in a second, we're going to go in and look at individual chromosomes. So here's chromosome 1, 2, 3, 4, and so on. So you can see that there are, in many cases, there are two discrete regions that correspond to the two copies of the chromosomes. And at the very end, we'll end on the chromosome X, and this is a male cell line, so there's only one copy of that chromosome. And this data, basically, you can look at the pairwise contacts between different loci. So you can see that matches actually high-C measurements reasonably well. You get these TAD domains. But if you actually then look at where the transcription active sites physically are present, they are present actually on the surfaces of individual chromosome territory. So in the yellow here is showing the chromosome paint done by DNA fish, or the red is the intron fish. So we see this almost in every chromosome we look at, that all the active sites are on the surface of these chromosome territories, and all the inactive stuff is in the center. So, and we see that, between different chromosomes, there's a lot of intertwining of the transcription active sites because, these things happen on surfaces, and they're basically in the space in between the chromosomes. So the overall picture is this, that basically the active sites looped out. Whether they do this as a function of active transcription or they just structurally loop out, is still to be determined. But then there's a lot of this compact stuff that you detect. These are sort of the tag domains that people see in these high-C measurements. Okay? So beyond looking at chromosome structures, we can take this data and then look at how the nascent transcriptome allow us to define cell types or cell states. In this case, each one of these dots is a single cell we measured. And then so each one of these is a 10,000 dimensional vector, right, with the amounts of the introns are expressed. And when you do this PCA analysis, you can see the mouse ES cells all clustered together. These are actually two separate replicates, and you can see that there's no batch effect, basically, in this experiment. ES cells in a different condition cluster separately, as well as fibroblasts. So one thing that we found that was really somewhat surprising was that if you look at it in different cells, the total number of transcription active sites are highly heterogeneous. Right? This suggests that some cells are actively transcribing, so they're making a lot of new transcripts, whereas some cells are essentially not as active or more quiescent. And so the question is, right, are these heterogeneity static in the sense that these cells will keep making a lot of more transcripts, and these cells will stay more silent, or are they actively interconverting? And then, so this is on the side we're showing you the actual distribution of the number of active sites. And then this is just based on simulation that, if you assume that each active site is essentially flipping a coin independently, then you should expect a much narrower distribution, right? So this suggests there's some extrinsic noise that synchronizes many of the active sites at the same time. So I'll go quickly. And so we've done post-trace experiments to measure how much the activity fluctuates. So we can incorporate a modified uracil that gets incorporated in nascent transcripts. And then we can measure both this activity as well as the introns to tell us the nascent activity sometime in the past and sometime now. So if you just wait 15 minutes between the two measurements, it's highly correlated. If you wait an hour, they become decorrelated. But surprisingly, if you wait 2 hours, then they actually recorrelate. Okay, so this is just plotting it out, higher time resolution. So there seems to be a 2-hour oscillation in the global activity of transcriptomes. You see the same thing, not just in ES cells, but also in fibroblasts. But then it goes away if you treat the ES cells with this 2I condition, which are 2 signaling inhibitors. And then we can show that if you measure the MRA at the same time, you can find that the intron activity is highly correlated with HES1. Actually, in fact, it clustered right next to each other, as well as FGF and SOX and STAT. So this actually corresponds to the observations that HES1 oscillates around a 2-hour period in many of these cell types. And in fact, work done by Wolf-Rex group, I don't know what the citation was showing, recently shows also that DNA methylation oscillates on a 2-hour clock in mouse ES cells. So I think that this 2-hour clock is something that may happen fairly globally. And in fact, the fact that it's happening in a desynchronized fashion between different cells means that you can't detect it using ensemble measurements. You have to do single cell measurements. And also that if you measure messenger RNAs, which has a lifetime of 3 or 4 hours, it will average out those fast 2-hour oscillations. Okay, so we can apply this kind of technology that we developed, right, to massively increase the multiplexing in single cells to memoir. So Jay mentioned this. We can generate mutations in cells and then decode these mutations using seqFish to read out the lineage informations. But then after the lineage tracking experiments are done, we can perform the seqFish on additional genes. So in the proof of principle experiment, we only did 3 genes, but now I'll show you we can now do 10,000 genes and then inferred rates. So this is good for not only measuring the nascent transcriptome and RNAs, but also to decode many of the lineage barcodes that we would need to generate these long lineage trees. And then we were going to work on integrating the seqFish data with the single-cell RNA-seq data that Jay showed you. Looking at our preliminary data, I think these 2 data sets will match very well together. and then we'll integrate it with Memoir in collaboration with Michael and Carlos at Caltech. So to echo what Jay said, I think Michael and I started working on this after the initial call from the Allen Foundation came out, and then there was this great interaction between Jay's group and our group, and Alex Shear's group together in the Allen Discovery Center. So thank you guys very much.
Are there any questions for Jay or Long Kai? Mark? The microphone's coming right to you.
So Jay, I mean, the one downside you mentioned with your approach is the rather shallow coverage that you're getting. And I'm wondering, is there anything you can do about that? Because, I mean, besides spend a fortune or whatever, you really want to look at variable genes, not just genes, right? And when you do your t-SNE plots, you're probably just looking at genes that vary from one cell to another, or you're just looking at all genes at once.
Well, okay, so I think it's a trade-off. I think my own perspective on it has been that, at least in these data, that the relative shallow, for the purposes of delineating distinct transcriptional states, the shallowness has not been a significant disadvantage.
No, I agree with that. I mean, that you obviously have gotten enough diversity.
And there's a ton that you get out of the time dimension, right? Just by doing multiple time points, even the heterogeneity within one time point starts to stretch out, and you can see how it connects to earlier time points, and resolve a continuous pseudotime similar to things you've done, right?
Right, but if you really want to know what constitutes the transcriptome of each of these or any of these cells, then you have just some information, enough to distinguish those cells from other cells. So my question is, since a lot of the RNAs, the expression is probably not varying very much from cell to cell.
You said is going to vary, or
it was not going
to vary.
It's not going to vary. I mean, ribosomal proteins, major cytoskeletal proteins. Is there any way you can hybridize out those transcripts before you actually do the analysis, and then enrich for the kinds of genes you'd be most interested in, not only in distinguishing different cell types, but also in telling you something more about what constitutes the interesting transcriptome of those cell types.
Yeah, so probably we haven't tried that on mammalian cells, but there are other organisms where we've had greater problems with ribosomal and other sequences dominating the data, and so we're actually trying some approaches like subtractive hybridization or CRISPR-based approaches to deplete some of that stuff, but we haven't actually gotten to the end point of that experiment yet.
Okay, because you're counting a lot of stuff that's doing you no good there.
Totally, yeah.
Right.
Any other questions?
I have one for Long. That was great. So can you comment on differences between hybridization efficiency of your FISH probes compared to like the mRNA and the introns?
Yeah, I think that mRNAs and the introns are pretty similar because they're all single-stranded, they're all RNA. So I don't think there's any significant differences. Yeah.
Okay, thank you so much.